OK, parfait.
The most critical for my science is to take a few minutes after, say, and say, what's in this for me? In a very selfish way, what can I use from this paper? And I'm really selfish because I'm actually thinking of every project in my lab quickly and going through what can I use.
Welcome to the Night Science podcast,
where we explore the untold story of the scientific creative process.
We are your hosts.
I'm Itai Yanai.
And I am Martin Lötscher. Agnel Sfeir is a leading cancer researcher in New York. She did her undergraduate training in biology at the American University of Beirut, after which she started a stellar scientific career in the US. She's obviously a big fan of New York City, because after postdoctoral fellowship at the Rockefeller, she joined the Skirball Institute at NYU School of Medicine as an assistant professor. There she was actually a colleague of Itai. But a year ago, she became a professor at the Memorial Sloan Kettering Cancer Center. Agnel received many awards, including the NIH Director's New Innovator Award.
And in her work, Agnel studies fundamental aspects of the biology of the cell. She tries to figure out the secrets of how information present in the chromosomes is maintained. To do that, of course, you have to be very creative, and she has been. She works on telomeres. These are the ends of our chromosomes, and mitochondria, the powerhouses in our cells. And she uses these two very different systems to study how DNA gets repaired, which is a crucial prerequisite for complex life. Last year, she even published a very interesting Nature paper about how mitochondrial RNA provides an intrinsic immune surveillance mechanism for cells in how they cope with double-strand breaks in the mitochondrial genome. Agnel, we're so happy to have you here. Welcome.
Welcome.
Thank you, Itai. Thanks, Martin. Well, I'm truly honored to spend an hour with you. And I have to say, it's fun sometimes to step away from the science to think about the process of how we do it. So I look forward to our interaction.
Yes, so do we. And it's all about the process, what we do here. This is really what we want to understand. And to get started, are there any aspects in that early training or maybe later in your PhD that you think influenced your creative development?
I would say it's a culmination of many things. So what is creativity in a way, right? It's trying, on one hand, to connect the dots between things that seem unconnectable, things that are a little bit far, but you need the idea or the mindset or the tool that makes that connection. So how do you get to that process? There's no secret recipe. It's like haphazard. But also to get to that, you need some persistence, some resilience, because the first experiment might not work, the second experiment might not work, and you have to keep going at it to get that connection, right? And then some fearlessness in order to go into places that no one has been before. You need that attitude. And I would say maybe the fearlessness and the resilience in my personality, sadly, though, I could attribute to growing up in a war. So I grew up during the civil war in Lebanon, and till I went to high school, it's just like a matter of trying to survive, right? And surviving a war makes you think there's nothing else. You don't fear much, right? So I would say my childhood gave me that fearlessness and the resilience that not only helps my personality, but also helps my science tremendously. Now, fast forward to my PhD advisor, Woody Wright, who passed away a few years ago, he was a wizard of molecular biology. He would take these simple questions and design himself in his lab that's in the garage of his apartment. He would design these incredible molecular biology techniques to answer these questions, right? And he had this incredibly creative way of looking at biology, influenced me quite a bit. And also I think the creative process has to be rigorous. And rigor in science is something very important. And I would potentially say I learned that from my postdoc mentor, Titia DeLange at the Rockefeller.
You said that when you're working, you need persistence and you need a sort of fearlessness. Some of us have this feeling when we're working on a difficult problem that we're just not sure if this is the time to be persistent because, like you say, you have to be, or maybe it is a crazy idea. So do you have like tricks for how you figure out whether the problem really is good and it's worth fighting for, or maybe this time they're right and we are crazy?
Yeah, right. That's the struggle. When should you stop? When should you give up? When are you
Right.
giving up not too early? And when do you give up not too late? I feel it's good to set up these time frame at the beginning of the project to say, here's a crazy idea. I am going to give it 6 months or 1 year, depending how long it's going to take to develop the tool to answer it. And just have that really strict time frame and say, I am going to keep going after 6 months or I'll give up. And sometimes you don't really need to give up forever, right? You often shelf things and then you come back to it in a year or 2 or 3 when you learn something new or there's a new tool that's out there that will help you answer it. But I wish I had this like magic wand to know when is a good time, but I feel it's healthy to set up that time frame from the offset. You say it's 1 year or 6 months, and after that, I will reevaluate the situation and decide either to move on or not.
Yeah, I mean, I can tell you that I can very much relate to shelving a project and maintaining this sort of loose end. You know, I think many of us have many loose ends that are just waiting for us to hear something at a conference, and then we pick up where we left off and have a new inspiration for it.
So you said something else just now when you were talking about your creative development. You said that creativity requires rigor. And that to me is a surprising statement because, you know, the way Itai and I tend to look at it, it's that the day science, the not so creative, the rigid part of science, that's where you have rigor. But in the night science, where you're creative and when you're just trying to come up with new ideas, new explanations, it's the opposite of rigor. So how do you see that? You seem to see it differently.
I don't know if it's different. I would say, like, you've got the initial idea, right? This question, because essentially everything starts with a question, right? Mm-hmm. And then the next step of the question is to find that initial observation that will tell you where you're heading is right or no. So it starts with the question, then you go to the observation or the finding, the initial finding. But then at that moment, that finding needs to be challenged. And the rigor is in this process of challenging that observation. So you have to go through a period of skepticism where you're saying, this is my interpretation, but there could be 5 other interpretations, and I need to rule these 5 other interpretations out before I hang on to my own favorite interpretation, right? So yeah. You're disproving this, and this is how I feel like creativity without rigor has to follow that creative idea.
No, totally. I think then we're actually on the same page because, you know, the way Itai and I would look at it, I think, is that you're constantly popping back and forth between the wild creativity and the rigor that kind of evaluates what you came up with, right? So you first come up with your favorite explanation, and then you also have to be creative to come up with the 5 alternative explanations. And then it's time for rigor to see which of those actually make sense in light of what data is out there.
Yeah. And then you follow the data.
Yeah.
So, Agnel, maybe you could tell us a little bit about a typical project unfolds in your lab, preceding the rigor, how the ideas come about.
It all starts from a question. I have a simple mind. I don't like complicated questions. I always gravitate towards simple questions. For example, I always laugh, as a PhD student, my PhD project was to answer the question of what is the last base of human chromosomes. The telomeres are TTAGG. Is it a T, the A, the G, or a G? So it cannot be more simple than that. And you could argue such an esoteric question, but at the end it turns out there is a machinery in the pathway that ensures the last base is always specific. And then if that pathway is manipulated, you lead to chromosome dysfunction. So I always gravitate towards simple questions. And this is in my lab right now. For example, how do the mitochondria talk to the nucleus? Or what happens when the mitochondrial DNA is broken? Or random nucleotides when repair happens. So this is the question, right? So we work hard on that simple question. But simple question, which means it's fundamental and simple, means people have not answered because it's difficult. Mm-hmm. So then we go through a process in the lab of trying to identify how we're going to answer it. And for example, if it's a postdoc project, there's often hours and hours in my office on the board. We're trying to plot different ways, different explanations. And some projects are easy in the sense you can come up with a hypothesis quickly and you go and test it. Others are not so easy. You can't come up with that hypothesis. And so you need to wonder a little bit. And wondering could take either you do a genetic screen, or you do a mass spec, or you have to do some wandering around until you get that handle. From that handle, you come up with a hypothesis, right? And then you test it and you come up with others and you test them or prove them or disprove them. But I feel the most fun part of the project is this process of wandering around to get that handle that gives you the critical piece that will show you how that project is going.
That's really interesting. Actually, you mentioned that you tend to gravitate to the simple questions. In our podcast conversation with Steven Strogatz, he said that many mathematicians see simplicity as a very important aspect of beauty in mathematics. And I agree. I mean, if you have an unsolved question that is simple to ask, that's the most fascinating things. I'm going to ask you to extend a little bit on what you just said. You said that many questions, it's not obvious what would be the hypothesis that could answer the question. So you first have this wandering around, this exploratory phase. And you say that's the part that's most fun.
Yes, because this is the part that you just don't know where you're going, right? Like, there's something fun about this uncertainty of which way you go. And just letting a screen take you is actually nice, following the data. This is really following the data, right? You run the screen and the screen highlights a pathway or a complex, and you just spend weeks and months reading about it, learning about it, and then going that path. This is the discovery, right? This is where it's nice to go into unexpected areas. And the screen lets you do that. While in a hypothesis, you don't often go wander into the unknown. You always gravitate towards what you know.
Yeah, I mean, that's what the discoverers of former centuries did, right? They just went somewhere where they didn't know what to expect, right? And that's the exciting part. I mean, that's how you make discoveries.
That's the fun part of it, in a way.
Yeah, and yeah, when you're looking for the unexpected territories and you're working with postdocs and grad students, do you find that sometimes, you know, they get a little nervous that they feel like their project should be on more concrete areas, and perhaps they join the wrong lab because this lab is wandering around? How do you sort of convey confidence that something will be found?
Oh, yeah, that is the crux of the thing, right? You know, you're a junior faculty, or maybe till recently, I'm a junior faculty. You're living with the uncertainty yourself, right? You're like questioning, well, am I doing things right now? Then you have these people who join your lab and they trust you and they put trust in you. And you're always questioning, am I deceiving them, or are we going to the right? Am I right, right? You're always questioning yourself. But I feel at the end of the day, one thing that does the trick to me, at least to me in my lab, is that if your lab members know that you are equally entrenched in that uncertain phase, like when they get the screen or the data or that pathway, I am totally immersed with them in learning about it, questioning it, figuring out the pros and the cons. And I think that gives them some confidence that they are not alone in that process and that we are a team in it. And that gives them a little bit more confidence in the process.
You know, I know what you mean, and it's so different from the way I've heard the liberal arts scholars do it. You know, there you do a PhD and you're really alone. You just maybe meet your mentor, advisor, I don't know what they call it, I think maybe advisor, once every couple of months, but they're not really with you. I think that's what's very unique about the sciences, that it really is sort of a common adventure that you struggle together.
Totally. And you know, actually, what I feel sometimes works for the lab is to have small teams. In my lab, we do telomere biology, which is what I was trained as. We also do DNA repair, and we do mitochondria DNA. So it's kind of diverse areas of research, even if the common theme is the chromosome or the DNA. That's the common language across all three. But one thing that my lab liked a lot was that we have these smaller mini teams, right? A mito team, a telo team, and a DNA repair team. And so I meet with people individually, but I also meet with these smaller teams. And there is a lot of sharing of the struggle and the joy of the discovery. And I feel that also gives them confidence that they're not alone. And my lab enjoys that.
Yeah, I'm curious, those teams, how big are they?
It's 4 or 5 per group.
Okay. Do you think that's an optimal size in order to do this exploratory phase where you're wandering around and trying to figure out what's going on? To have 4 or 5 people?
Yeah, because it's one thing, it's you, or it's me and the student, but it's different when you have another postdoc or a more senior member listening to that thing, right? So when they're presenting their finding or they screen, and then you have more than one person reaching the same conclusion, they're more confident in taking that second step. Does that make sense?
Yeah. That makes a lot of sense. But, you're talking about basically the social or sociological aspect of this team. But when you think about coming up with new ideas, new directions, maybe in the end coming up with a hypothesis, do you find that's easier in such team discussions, or is that something that happens more in the one-on-one discussions that you have?
I think it's both. It can come from both. Like sometimes we do come up with amazing ideas in our smaller team meetings. And you have this, oh yeah, I have not thought about this way. This is nice. And often it comes from one-on-one. Like we always joke, like usually when we have these 2-hour smaller team meetings, it's the last 5 minutes where the best ideas come out. Like you've got an hour and 55 minutes where you're going through the data and like talking and back and forth.
Right.
It's the last 5 minutes when you nail it.
Why is that?
It's so true. Yeah.
It's like as if your mind needs that 1 hour and 55 minutes and going around. And it's like when you know it's about time to wrap up, then you're like, okay, here it is.
Yeah. And I think the mistakes people make is when they reserve just a short time for a meeting, maybe an hour and a half, and then they never get to that part, right? Because you sort of have to incubate all that, and then you get the payoff at the end. It's also when a speaker doesn't leave some time for questions at the end that you have this unfortunate problem of not getting to the insight, because it's usually when someone asks a question.
But Agnel, I would like to understand this better. Do you think it happens when you kind of run out of the discussion and then there's not really much more to say, and then suddenly you realize, oh, that's the answer? Or is it more like you look at the clock and you see, oh, the meeting has to be over in 10 minutes. Well, now we really need to come up with the finding.
Oh, it's difficult to answer because I honestly don't know, right? But it always happens and I don't know why. It could also be because you've got your time to listen, and listening is important. Listening and looking at the data, and we're all looking at the same data, and go through the different explanations. But then you've got at the end where you synthesize everything. And your mind is programmed to synthesize everything. And before you leave, you have to reach a conclusion, right? And then maybe it's a time frame, or I don't know. But for me, it always happens. The last 5 minutes of our meetings is when the best ideas come up.
Wow, maybe it's because there's like this processing time when you're listening to a presentation. You're just taking in the information. And like you say, the synthesizing only happens when you can just stop bringing in new data and just think about what you've heard. And then you need to kind of allow for that.
And just like going back to this creative process, right? Again, creativity is not a very objective term. Different people view creativity differently, but like, how do you get there? And one thing I started telling my lab, and I've been implementing for like 4 or 5 years now, this idea of a selfish behavior when reading a paper or listening to a talk, right? Because often you read or we do paper for journal club, and the first thing trainees want to do is tear it apart and go over every band on the gel and all of that, which is fine, right? And then you go through a phase where you say, okay, what did we learn from it? Or from a talk, you say, okay, what's the take-home message? What am I going to remember from this talk? But then I feel the most critical for my science is to take a few minutes after, say, and says, what's in this for me? In a very selfish way, what can I use from this paper? And I'm really selfish because I'm actually thinking of every project in my lab quickly and going through what can I use where? Like, is this tool helpful here? Is this way they did the experiment going to help this person in my lab? And I kind of am training my team after every journal club to spend 10 minutes thinking selfishly. Like, what's in it for us? And I feel that also helps because it's a way to generate ideas that will allow you go through this creative process through any project.
Yeah. I think that's a great strategy, actually. I've never done it that way consciously or as consciously as you describe it here. But I think that's something I'm going to introduce in my group as well. Like you're saying, it also gives you a much more direct connection, I think, to what you learned from that paper. By connecting it to what you're doing, it becomes much more relevant, and you're probably also more likely to remember what was done in that paper.
So basically on my computer, I have a file notes called Selfish 101.
Okay.
And I often write down what are these selfish conclusions that I reached from every talk or paper.
And do you sometimes go back to that file and pick up something that you didn't use initially?
Yes, I do look at it every now and then. Because sometimes you remember something, but it's vague. So I go to the file to read what were my notes back then to see what was really the idea.
Agnel, I wanted to ask you about the creative process from the angle of the visual aspects, because I think in working on the biology of the cell and in thinking about chromosomes, I think there are so many ways to imagine that. Like, do you find that, for example, when you said that you're on the board and drawing, is a lot of that images of the cell and you're trying to kind of in your mind have a super resolution microscope where you're trying to see things, or is it more logical and less on the visual side?
On the board, it's probably more logical, but I like to see things on the microscope. It's when you see it, you just have a different idea of what it is. I don't do experiments anymore in my lab. The best experiments I do now is probably a mini prep every now and then. But if someone is on the scope and is like, I know they're doing an experiment, I don't mind just poking my head into the scope room and looking at the cells or the chromosomes or what they're looking at. And that sometimes you see things that you're not expecting and it makes you think differently, right? And especially if the students are new, their eye is not trained to look for these weird things that could lead to a project. And so by sitting with them on the scope and looking at the screen on the scope, you just try to point out these weird observations to them, and you kind of train them to look for weirdness in the slides.
Also you mentioned that you don't do experiments anymore. So did you find that the transition from having like first-hand experience and seeing everything directly to now being more one step removed in the sense that you're depending on the members of your lab to bring data to you? Was that like a smooth transition?
No, I feel sometimes I'm so annoying to my lab because when you're like so excited about something, I go to their bench and it's like, have you done it? Have you done it? And it's a fine line between annoying the heck of them or showing them I'm genuinely excited, right? And some people tell you, leave me, don't come, I come to you. And the others are more open and they show you the gel or they show you the data quickly. But just the idea of I don't have the time to do an experiment and to run something from A to Z is a bit annoying, and it is frustrating. As I said, my PhD advisor was running experiments every day and he would often start them in his garage. So it's not easy. And I attempted in the past few years to start a project, but I often halfway through I'm not done and someone else in the lab has to finish it, and they don't like that much. So they basically nicely told me not to run experiments on my own. But I helped them. I'm good at mini preps. Yeah. I can run mini preps here and there when I want to be in the lab.
So this is really interesting how you talk about your supervisor who continued doing experiments, that you stopped it and you find it frustrating. Do you also feel that stopping doing the experiments yourself has changed anything about your creativity?
I'm not sure because, when I was a postdoc, I was working hard for 9 hours a day. And then when you're running the experiment, you're thinking mostly about the details of the experiment, the design of the experiment or the method. Your mind is not wandering much, except when you're done at the end of the day. And like after dinner, I used to love these time between 10 and 1 in the morning when I'm just analyzing data and I'm interpreting my experiments. I feel this is when I really thought about science. While during the day, it was mostly thinking about the experiment and not the science behind it, right? But now I feel when you don't do experiments, you're constantly thinking about science. And I like it. Like I'm thinking about our projects when I'm walking or taking the subway or whatever, all day basically. And I like that.
So you feel it gave you much more space for creativity?
Yes. Sadly, yes.
Why do you say sadly?
Because sometimes it's nice to be in the lab. I enjoy being with my team. There is some satisfaction to doing an experiment. There is some satisfaction. This is what we're trained, right? Then all of a sudden you leave it. But also, it's just impossible at some point to keep doing experiments or to do them properly, to do experiments as well as you did when you were a postdoc.
Agnel, do you think it's impossible to do experiments? By the way, I can totally relate. I also don't have time to do experiments, although I still code a little bit. But do you think it's impossible because you need to do night science and all kinds of thinking for so many projects now, not just one project as when you were a postdoc? Or is it because there's something about academia that forces us to be kind of like administrators? And
I think it's both. Because on one hand, now you're responsible for not just you. You're responsible for like 10, 12 people. And you have to think about their projects with them. At least that's how I operate. I cannot just be handed a paper 3 years after a project and be told, think about. I want to think through the process. So on one hand, you need to think more. So you can't really do these 3-day-long experiments. But also, you're running between meetings during the day and like your calendar is sometimes overcrowded and you don't have this 7 hours to be on the bench uninterrupted. I tried, but when you realize your experiments suck, this is when you think, I better do what I'm better at. It's in my office.
So earlier on, you said that some students, when you walk into the lab and you ask them, how is it going? They just tell you to go away and don't come back, right?
Yeah.
And then others more appreciate you being there and worrying with them about the outcome of the experiment. Do you feel that the different people you work with also differ in how you can be creative with them?
Yeah, because some people, you need to give them the space and they come up with the ideas and your job is to challenge them and help. But others, you have to feed them very subtly, give them the hints, and then they can build on that. Does that make sense?
Oh, that makes a lot of sense.
Like I've got students who basically ran their show and they come in and they tell you the idea and your job is just to challenge it, right? As we said, every creative idea needs to go through a phase of being challenged. And so in some cases, my job is I serve the role of the person who's challenging my trainees' ideas. In other instances, I'm the one who's slowly guiding them and we're working on it together. And I think that the trick is how do you learn how each person works? You need to go through a phase where you learn about every person in your lab, what makes them tick and what's the process for their own creativity. And that is not easy.
Do you have any tricks for how to do that not so easy task?
I wish I say I always succeed. I fail more than I succeed sometimes, but I feel this needs time. You need some time to learn about each person. And this is why when someone joins my lab, we have these weekly meetings of at least an hour where we're discussing the project. And as the discussion is going, you learn how every person think and how they like to think or how they like to be creative.
And when they bring an idea for their project, how do you know if that's the one they should work on? How do you choose your ideas? Maybe they don't fit the style of the lab. Maybe they don't resemble a simple question the way you like it. Or maybe it's an exciting new direction that you should gamble on. How do you decide?
I think if they're able to convince me and the team and that mini team about the question and how they're going to go at it, I support them. There are instances where I didn't feel that was core to my lab, but this is where they wanted to go. You just have to go with them, right? Our job is not just to do our science, but to allow them to do their science. And they have to go where they feel like they want to go. But again, it has to go through their phase where they convince me and the rest around that this is really a worthwhile idea. They have it figured out. They have the plan. And then they just go.
Yeah, I think, of course, you're right. Science needs people to be excited about what they're working on and to work on their own ideas. But on the other hand, you as a very successful scientist, you certainly have a good intuition about which problems are really interesting enough to spend a lot of time working on them. And that's an intuition and also an experience that many of the people in your lab probably don't have. So don't you think it's also part of your job to guide them in this respect?
Absolutely, right? And so the question is, how do you teach them to go after worthwhile questions? What is the measure of a worthwhile question? To me, at least, what I try to say, the more filling in the gaps, the less interesting the question is. The filling in the gap is a good measure of how limited the question is. And I try to gear them towards find these projects where you're going to be launching something new that will lead to 10, 15 years of discovery, and not just a project where you're filling in the gap and the project will end with the discovery that you make. But this is me, right? This is very personal. Maybe other people view projects differently, and some people do gravitate into filling in the gaps, which is important. It's important for the process of discovery. But for me, I kind of gravitate away from filling in the gaps, and that's what I try to tell my lab.
Sometimes in the process of filling in a gap, you could reveal a whole new world, and it might surprise you, but I know what you mean.
Absolutely.
Yeah, but there's different tastes in science of what people find interesting. So if you're not so interested in filling in the gaps, but there's other people who love doing that, then
totally. No, absolutely. Yeah.
Yeah.
And it is important to fill in the gaps, right? And it's important to have those people who want to address these questions. But it's all personal at the end. It's personal preference.
It's true. There could be some people opposed to you. They might like a very complex problem where there's so much that's already been done, and they do want to fill in the gap because they like a tidy universe.
Totally.
And yeah, you, as we mentioned in the introduction, you recently changed environments. You were in NYU and now you're at MSK. And I'm wondering how this change of environment is playing out for you. Are you forming new collaborations, and is that shaping your creativity?
It is invigorating to move, actually. I spent 9 years in the Scribble Institute at NYU, and they were like fantastic. I cannot tell you how much these years shaped my science, shaped me as a scientist, as a mentor, as a colleague. They were incredible. But admittedly, moving is invigorating because you learn about new science. People at MSKCC are doing fantastic science, like so interesting, so deep. And you find new collaborators. We've got a number of new projects that we started. So I don't want to be preaching people to say moving is good, because sometimes it's not. It is annoying to move, but it is also invigorating. And it's challenging, but in a good way, because you want to learn about the new stuff that's going around you. Because at some point, being in a place for so long, if you predict what your colleague is going to be talking about, so you kind of zoom out the first 5 minutes of every talk. But here you can't now. You have to focus from the first second because you don't know what they're going to... Yeah. And there's something super nice and fun about this.
So, Agnel, we talked a lot about creativity, and you told us how you love this exploratory phase where you're trying to get to the bottom of your question. Do you have any tricks or any conscious methods or habits that you use when you're in that phase, when you're trying to find what could be the kind of answers for this question?
The first trick is be patient. This is something like I always tell my lab, because the person who's working on the project in that exploratory phase, they're frustrated, right? They're scared, they're frustrated. What if nothing pans out? So first thing you need to just be patient. It's going to come. And then let's not jump the guns. Let's analyze the data when the data comes, right? Our exploration is through a CRISPR screen, right? And once the screen is analyzed, then it's like complete obsession, I have to tell you. Like for me and the person, it's like nonstop. I would say as long as I'm up and awake, I'm thinking about the results and the pathways and reading and going back and forth. I do have some like obsessive personality. When I'm working on a paper or I'm analyzing the screen, there's no day and night. It's just one. Right? Mm-hmm. And that phase is the same. You're just constantly thinking about the data and where you're going and try to come up with a hypothesis, or often more than one, to answer, to see where that leads you. Just be patient. That also, as soon as the screen is done, is complete, utter obsession.
Yeah. Tell us more about this The obsessive phase sounds remarkably interesting.
I'm sure you guys go through this. When you're writing the paper, it's not that you're thinking about it 9 hours a day and that's it. You're just thinking about it nonstop.
You dream about it. You have night science.
You wake up in the middle of the night and you think, oh, what about this? And you often have to grab the phone and write it so you make sure you don't forget about it in the morning, which is more borderline crazy, I would say. But this obsession, every project has to go through this, or mentally you have to go through the obsession phase. It's only fair for the project to obsess about it, right? It's just, there's beauty in that. It matures differently in your mind.
I think if the project does not have the obsessive part, it really is getting cheated. That really separates maybe scientists from people working in, I don't want to name any other jobs, but ones where you do a 9 to 5 and then you go home and you could have some hobbies. In science, like you say, we have to dream about it. We can't put it down. We wake up with a start at night. So I think that's the difference, that the project needs you.
Yeah, and even if you have hobbies, right? And like on a bike ride or what else, sometimes you just start thinking about science. I always give the funny example. When you live in New York City, New York is so crowded and so busy and so loud. But somehow, when I'm walking, I create a bubble around me. I put my headphones. I just have no idea what's going around. And you might be surprised, but I'm thinking about science on the streets of New York City often. It is dangerous, but it's...
Okay, but look both ways when you cross the street. I know what you mean. You have to kind of be in a bubble and be on this alternative plane where you're thinking about the project.
And of course, this just shows how lucky we are to be allowed to work in science, that it's so exciting and it's so... You obsess about the questions, right? You do it because you have to do it, right? That you want to do it. I think that's a huge aspect of what makes working in science so attractive.
Yeah, it's your question, right?
Yes.
It's not a typical job at all. I remember when I read The Selfish Gene by Richard Dawkins, I got so interested in biology, but it was only when in graduate school, in the first semester, like you said, I had this obsessive part of a project and I couldn't go to sleep. I was thinking about it nonstop. And that's when I fell in love with research. I think there's a difference between liking science versus liking research. The research has the obsessive part. So one question I have for you is, what was your first obsession, Agnel? Do you remember the project?
Yeah, it was a master's student at the American University of Beirut. We were trying to study the crosstalk between cell-cell interaction and cell-ECM interaction to see if the way the cell interact with the extracellular matrix can influence how the cells talk to each other. And I was supposed to do just 3-month rotation almost. And then I just couldn't let go. I stayed for basically 2 years on the same and ended up doing my thesis on that, right? I feel maybe that was my first true introduction to doing research. And this was my first obsession. And then since then, countless.
Well, like you were saying before, that the insight of a group meeting tends to come only in the last 2 minutes. I feel like we just had a breakthrough here with this notion of becoming obsessive about a project. It sounds unhealthy in some respect, right? To have an obsession, but that's just how we scientists roll, I guess.
Some things are not always healthy, right?
Exactly.
Maybe science is not good for you, but at least it's fun.
Yes.
So I think we covered a lot of ground today, Agnel.
Thanks, Itai.
Yeah. Any final words about
Any insights that you gained in the last 5 minutes?
Apart from knowing that I'm an obsessive person.
Yes.
I'm obsessive addictive personality, right?
But in a good way.
Yeah.
But in a way that also benefits society. So
Yeah, I have to say, we don't have a job. I think science becomes not fun when you start viewing it as a job. That's
Yes.
Right.
It's who you are. We identify being scientists before being from the US or Lebanon or what, this is our identity basically. And a part that I really enjoy is like the teamwork, the idea of you work with 10 or 12 people who are fantastic, they trust you, you trust them, and you build this relationship that's actually for life. And starting a lab is not the easiest thing. You just come to it being clueless about so many things about how to run a lab. But I always feel, at least to me, when I started my lab, I thought one thing I'm not going to worry about, and that's tenure. I'm just going to not be obsessed, because people start their labs and thinking, oh, I need this to be tenured. I need this to be tenured, right? I feel that is the utmost mistake. And maybe I'm wrong, but that's me.
No, you're right.
Science is hard sometimes, like, there's so many things. I should not be obsessing about tenure. And if it comes, good. If not, there are many other things in life that are amazing that we can do. And just enjoy the process without kind of worrying about tenure. And I implemented this. I didn't care much about the process. I just wanted to have fun in what we do and have my lab answer questions that are fundamental, not small ones that are filling the gaps, as I said. We didn't fail completely, so that's good.
you didn't fail in the least.
That was beautiful. Agnel. I appreciated that a lot.
That was a fantastic summary. Agnel, thank you so much for this discussion with us.
Thank you,
That was
Martin.
great.
Thanks, Itai. This has been fun.
Thank you.